Hello and welcome to another episode of Physiology with Shakti, and we'll be discussing smooth muscles today. So to start off with, we'll go to understand what is the structure of a smooth muscle, and then we'll go on to understand the mechanisms of smooth muscles, and all through the way we are going to compare and understand how different it is from your skeletal muscles or your cardiac muscle. Okay, so starting with the structure, you can say that a smooth muscle has a fusiform shape. And this structure has inside it multiple dense bodies. So these dense bodies actually have inside them something called alpha actinin. So if you know this already, alpha actinin is a structure which is tethering your Z disc to your actin actually in your skeletal muscles. So this is something that is there at the Z lines. So here instead of a Z line, we have the dense bodies which has an actin. Okay. Now these dense bodies are all interlinked to each other with intermediate filaments. So as you can see, they're all tethered to each other through the dense bodies all throughout. And these intermediate filaments, the most common ones which we have to talk about in smooth muscle is your vimentin and your desmin. So we have the vimentin and the desmin. Okay. Now beyond this, we also have your actin and myosin. It's just that they do not have the usual striated pattern that you see in your skeletal muscle. Okay, so we have your actin filaments, thin filaments, as well as your thick filaments, the myosin. Okay, so all throughout there is dense bodies. Interlacing them is your intermediate filaments, and interspersed between them, connecting them is also actin myosin complex. You have to understand the actin here has the tropomyosin, but it does not have troponin. So how exactly does contraction happen here? It is quite different from how it happens in your skeletal muscle. We'll come to that. Another thing is your myosin which is there. This is around 30% less as compared to a skeletal muscle. Okay. Now these dense bodies which are finely tethered near the membrane, plasma membrane, they are tethered using focal adhesions. So focal adhesion through integrin will bind outside to the extracellular matrix. So this dense body like this, and on the other side there is of course your actin and intermediate filaments, everything is there. Okay, and focal adhesion, integrin, extracellular matrix. So this is the fusiform shaped smooth muscle structure, the first thing we're going to be talking about. Now these structures exist in two forms. First thing is what is called a unitary form, and another is multi-unit. So what exactly is the difference in this arrangement? One, we have gap junctions which are present in unitary. So between them, if you zoom into it, between them there are multiple gap junctions which keep connecting between them, which will not be the case in your multi-unit. Okay, so we have gap junctions which are there. And even the nerve supply which is coming in, it usually comes in like this. We'll put a myelinated nerve, and it goes here and form what is called varicosities, like this. Okay, and this is not going to all the muscles, just maybe one or two, very few are there, but that would suffice for the entire thing because of your gap junctions. So these are what are called n% junctions. Okay, and they are less in number. On the other hand, if you look at a multi-unit, each of them will have their own separate n% junctions. Okay, so I hope you're understanding the difference between them. So in unitary, they all act together as a whole. There are multiple gap junctions. There might be lesser number of n% junctions, but because of the presence of gap junctions, it is transmitted throughout for it to act together. Multi-unit, each of it has its own separate innervations. They do not have gap junctions or have lesser number of gap junctions. Examples for this you can talk about. Unitary is your whole hollow viscous. So your intestine, your genitourinary tract, where the bladder is there, ureter is there, all of that. Okay. On the other hand, multi-unit examples are your iris in the eye, the ciliary muscles in the eye, the piloerectile. Okay, so these are examples of your multi-unit. So when multi-unit is stimulated, it is more precise because each of them is receiving separate innervation. Okay, but it does not last that long. It's more shorter but more precise. Okay, on the other hand, your unitary, they last longer actually. Okay, they last longer, less precise. Okay, so this is one thing that you need to understand about the difference between how the structure of smooth muscle can exist in the setting. Now that is clear. This innervation which is coming, the n% junction, will go and zoom into it. Okay, to understand how is a neuromuscular junction, or what exactly is this n% junction. So these n% junctions have multiple vesicles over here. You can also draw the mitochondria which will be there inside it. Okay, one thing you have to understand, there is no difference in the postsynaptic terminal. In skeletal muscles, usually the postsynaptic terminals are thrown into multiple postjunctional folds. Such a specialization is not existing in your smooth muscle n% junctions. Okay, that is one thing that you have to understand. Second thing you need to understand, how exactly is the depolarization wave coming in? Because in your skeletal muscle, we have the T-tubule structure. Okay, so we have a structure here which is a T-tubule like this, which goes in, and on either side we have your sarcoplasmic endoplasmic reticulum like this. Okay, so the signal goes in like this and it activates dihydropyrimidine receptors, ryanodine receptors, all that is taking place. Here, how exactly does depolarization wave reach over there? What is the steps? And how exactly does the depolarization wave look like? Because in a skeletal muscle, how would it look? It look like a simple spike potential like this. Okay, so how exactly will it look over here? So this is where you have to understand smooth muscles are peculiar in that sometimes a junctional potential can develop. These junctional potentials can be excitatory or inhibitory. And in fact, they are not action potentials. They are actually graded potentials, localized graded potentials. But somehow, in certain instances, a junctional excitatory potential is known to make a smooth muscle contract. So even without an action potential, you can see a contraction over here. One. Two, there can be a simple spike potential, like the one we saw earlier, a spike. Okay, and usually it's seen that some way sodium is involved here, but not essential. So sodium just helps it to make it faster, but calcium is the ultimate thing for a smooth muscle. Then we have plateau potentials, like the ones we see in your cardiac muscle. So these are especially present in your genitourinary tract smooth muscles. Why would you need that? Because of the presence of this plateau. During that period, there is longer period where calcium is coming in, resulting in a more maintained tone. So such plateau potentials are seen in genitourinary tract. The most common example that we use generally when we talk about smooth muscle is your slow wave and spike. Slow wave plus the spike potential. Okay, this you see in your intestine, in your intestinal cells of Kyle. Slow wave plus spike potential. How does it look? So your smooth muscles tend to have a slowly undulating RMP. It's not a fixed RMP like minus 90 you see in a skeletal muscle and all. Instead, it is slowly undulating between minus 20 to minus 65 millivolt. Okay, all of these values I am taking from standard textbooks only. So we have a slowly undulating RMP like this. Okay, so while it is slowly undulating, sometimes there can be an activation of a voltage gated calcium channel. Once the voltage gated calcium channel is activated, it can result in spike potentials. But these spike potentials, compared to that you see just with the sodium, will be much more slower. Okay, the time duration is longer. Okay, the voltage gated calcium channel is opened up. So on top of the slow wave, a spike potential will now come, because of which calcium is coming in. Once calcium is coming in, that will activate another receptor. Okay, so that is your calcium dependent potassium channel. So potassium channel just got opened because calcium came in. That potassium channel will result in repolarization. But while repolarizing, it will go into hyperpolarization. What does hyperpolarization do? Hyperpolarization will inactivate your voltage gated calcium channels. So normally this voltage gated calcium channels, unlike your sodium channels that you normally see, they take longer time to actually get inactivated. So now that voltage gated calcium channels are inactivated, there is no longer calcium to keep the calcium dependent potassium channels open. So then what happens? That will inactivate the calcium dependent potassium channel, thus making it go back. So now we have a whole repeating cycle. Then you have slow waves in between. Calcium will come in through voltage gated calcium channel, calcium dependent potassium channel, hyperpolarize, again it goes back. So this can keep happening. So this is the kind of potential that you see in your intestinal cells. So these are all different ways by which calcium can come in. Okay, So once this is clear, how exactly is the calcium coming? Because again I told you, we have dihydropyridine and ryanodine receptors which are there in your skeletal muscles. Okay, we have another thing over here instead. This is what we call a caveolin. And next to the caveoli, we have the sarcoplasmic endoplasmic reticulum. Okay, so we have the caveola, sarcoplasmic, endoplasmic reticulum. Now this caveola has a particular voltage gated calcium channel. So calcium channel which is voltage gated, which is present over here. Okay, through which calcium can now come inside. This is one way calcium can come in. Okay, what are the different ways calcium can come in? One, your voltage gated calcium channel, or you can just write it as CAV also. Two, this calcium which is coming in can activate another receptor which is ryanodine receptor. This is very similar to what you see in your cardiac muscle actually. So like that calcium can come out. Okay, or there is another channel over here which is IP3 receptor. So on the surface there can be different receptors which are there, to which ligands can come and bind. What are the different ligands? One, ATP can come and combine to particular P2X receptor channels. Okay, or your acetylcholine or your adrenaline, noradrenaline, all that can come and combine to G protein coupled receptors. So ultimately, let's just take an example of your acetylcholine, which will now come and combine a G protein coupled receptor is there. One of the things that activates downstream is your phospholipase C. What does phospholipase C do? It splits the membrane components to two things, IP3 and DAG. So this IP3 which is there, that is now going to go and stimulate the IP3 receptor, resulting in calcium to come in. Okay, so this is where you have to understand there is both intracellular as well as extracellular calcium, which is important over here. Because when you talk about your skeletal muscles, your intracellular calcium is more important. When you talk about your cardiac muscle, extracellular calcium is more important. If you talk about your smooth muscles, both in the intracellular, extracellular has a role. Okay, by itself can contribute. So extracellular through the voltage gated calcium channels, intracellular stores through the ryanodine receptor channels, or through ligand gated IP3 receptor channels. Intracellular stores can also be involved. Now there is a third mechanism. Okay, so we were talking about different things. One is intracellular stores like ryanodine receptor or IP3 receptor. Now there's a third thing which is calcium independent, or what is called store operated. So how does it work? Usually on the membrane we have a particular receptor channel called ori. Now this ori has a cyclic component to it, which is coming very close to your sarcoplasmic endoplasmic reticulum. Now, what is the whole function of your sarcoplasmic endoplasmic reticulum? It is your calcium reservoir. When the reserves drop and it reduces, one of the things it does is a particular protein structure will come on top, which is called a stem protein. So this essentially creates a medium for direct transmission of calcium from outside to now come inside and replenish your reservoir. Okay, usually this sort of a mechanism is present in things which do not have a caveolae, which has some other requirement like the lymphocytes, macrophages, epithelial structures. All of this usually have such mechanism, but it is also seen that in smooth muscles this also plays a role. Okay, this is what is called store operated or store operated channels. Okay, so three things by which calcium is coming in. One, the store operated. Two, the voltage gated calcium. Three, intracellular stores like ryanodrin receptor channel and IP3 receptor channels. Okay, now that is clear. Once finally calcium has come in, what do you do with the calcium? Because troponin is not available. How else will the muscle contract? This is what we are trying to understand. Okay, so one of the things is there are two proteins which are there inside, which are calponin and caldesmol. Okay, these structures normally tonically inhibit the interaction between actin and myosin. So what do they normally do? They normally inhibit the interaction between your actin myosin. So now the calcium which is going to come in will combine to these structures and inhibit this. Inhibit the inhibition. Okay. Now apart from this, the main thing that we have to talk about, I'll just draw you a myosin. So what we are seeing here is essentially your myosin heavy chain. Heavy chain has a head, a hinge and a tail. So myosin heavy chain is what you're seeing. Apart from your heavy chain, it has a light chain which is called a regulatory light chain. And it has an essential light chain. Okay, now we're gonna draw another structure which is myosin light chain kinase. This is a myosin light chain kinase. Okay, we'll just keep it there. Now what happens? The calcium which is coming in, where does it go to? This is where you have to draw another structure, calmodulin. This structure is what we call calmodulin. So calcium will come and bind to it at 4 different sites like this. Thus what will happen? So now there is a configurational change which is happening because of the calcium calmodulin complex coming to the myosin light chain complex, and this over here is the active site of this myosin light chain kinase. So what it does is this myosin light chain kinase will now go and phosphorylate a particular area in the regulatory chain of light chain of the myosin head. Okay, so especially in the 19th serine area, phosphorylation is going to happen. Residue which is 19th serine. Okay, that will result in changes whereby it makes it much more easier to utilize ATP hydrolysis and result in configurational change for the myosin, helping it contract. That plus your calponin and caldesmon inhibition is also lost. Okay, so these are the various reasons why calcium influx can, instead of through troponin, through a calcium calmodulin complex formation, and myosin light chain kinase can result in contraction. Now, despite this, there is another substance which is myosin light chain phosphatase. So what does this do? This does the exact opposite. Okay, it removes this complex, so this results in your relaxation. This is not only the only thing, apart from that, especially in your vascular smooth muscles and all, nitric oxide is also involved in various ways of relaxation. Okay, this is in the end how smooth muscle through calcium is going to get activated and contract. Now, there is some peculiarity in this whole activity. Okay, not like the one you see in sliding filament theory. Instead, we call it a latch bridge. We call it a latch bridge mechanism. What is different here? One, the frequency of your cross bridge is less, as in less than 1/10 time that you see in a skeletal muscle. So the speed with which the cross bridges happen, that is much more less. 2, detachment rate is also less, which implies that it stays maintained for a longer time. So it's like a latch you put and it'll stay there. Okay, so this is one efficient way by which it can use less ATP. With less ATP hydrolysis, this is able to attain high tension or force. You understand what I'm trying to say? Usually in your skeletal muscle, what is happening? Your cross bridge cycle, there is a actin, myosin, it forms, uses ATP, COX does this, it keeps doing it. Multiple cross bridges will keep happening. It's not like that over here. One latch and it stays there for a long time. Okay, and then later another one. So it's not like, it's not easy to keep going on. So frequency is much more lesser, detachment rate is less. Okay, so it's able to maintain it longer, and with less ATP hydrolysis it is able to achieve higher tension and force. Another peculiarity you have to know is the normal length tension relationship that you see is not there. And as a result of that, there will be something called a plasticity property over here. Okay, it's not there as in, it's not as linear as you'd think. Sort of unpredictable actually. You can't form a meaningful relationship between that. Okay, so in Frank-Starling's law, length is increased, force of contraction is increased. It's not entirely so in a smooth muscle. Okay. Then if you look at it, as I was telling earlier, around 30% lesser myosin is there, and around 100 times lesser ATP is utilized compared to skeletal muscle. So these are all peculiarities of the latch bridge mechanism. So I hope I was able to simplify the entire thing of smooth muscles, because smooth muscle is something which is not given that well in textbooks, and students usually study skeletal muscle, students usually study cardiac muscle, but leave out smooth muscle, and when a question comes they're not able to answer it, explain it, or understand the components which are involved in this.